Mr. Investor, welcome back to the channel, baby. BNGO is dropping like it's hot, but I'm still not worried, baby. In fact, I've been doing a little bit more research. You're not going to want to miss this video. I've done a lot of research this afternoon. Make sure you hit the like and subscribe button, and always remember, none of this is financial advice, baby. It's for entertainment only. So I'm on 9,800 shares of BNGO. I think I'm at $2.50 or $2.40, something like that as an average, but I'm looking to scoop up even more. I've been looking at researchers past and present and to see what they've been finding with the machine. Dr. Broach, for example, was using whole genome sequencing with Illumina's Novasec and optical genome mapping with Sapphire system from Bionano Genomics to explore the dark matter of cancer genomics. He wanted to reveal undetected structural variants in leukemia, and he noted that optical genome mapping provides otherwise inaccessible structural clarity of HPV-induced genome alterations. With Illumina's machine, he was saying whole genome sequencing is very good at picking up gains, whereas optical genome mapping is much better at picking up losses and insertions and inversions, and both technologies are effective at picking up translocations. This is a gentleman that received a Lifetime Achievement Award for his contributions in the field of yeast genetics and outstanding community service, and he received this back in 2016. Now, when searching optical genome mapping over on Twitter, I looked at Chloe and I could see that she works for Bionano Genomics in marketing. She was showing all the latest research from researchers around the world, showing their posters and what they've been using Bionano Genomics optical genome mapping to explore. With Bionano Genomics Sapphire, apparently Dr. Catherine Anognostopoulos's poster had detailed how Bionano Genomics optical genome mapping had precisely determined a breakpoint on a certain gene in a girl with developmental and epileptic encephalopathy. Sounds very hard to pronounce. It basically means damage or disease that affects the brain, and it can lead to an altered mental state, leaving you confused and not acting like you usually do. And when looking into the background of Catherine, I saw that she was a geneticist at Genomedica. What exactly is Genomedica? It's a diagnostic center in Greece. I then looked at a few other posts from Chloe, and we could see that Eddie Dubois, who is a researcher and a PhD student, had also put up a poster of optical genome mapping and how it could replace the current workflow for hematological tumor characterization. This was on April 20th, 2022. And if we pop open the abstract for their experiment, we could see what they believe. They're looking to replace current techniques for hematological tumor diagnostics with optical genome mapping. So basically with this experiment, they're saying that, it's quite expensive, some of these techniques, and time consuming to actually resolve complex rearrangements in leukemia. So they wanted to see if they can investigate whether Bionano Genomics optical genome mapping has the potential to replace the current methods into a single automated test. And when scrolling down, you can see in their discussion, they said in their experience, optical genome mapping is faster, less expensive, and of higher resolution than the current methods. So it combines the advantages of karyotyping, SNP array, and FISH, and it has the potential to replace all these techniques. It's also important to note that they're going to do a few more experiments before they introduce optical genome mapping into their tumor standard of care workflow. They also want to confirm potentially clinically relevant extra findings, and they want to map 10 more additional bone marrow aspirates. So they're going to continue experimenting. They want to confirm some information before they implement it into their standard workflow. Now, Chloe works at Bionano Genomics. She works in marketing, and she also shared another post showing that, it's been utilized over at Hospital Del Mar by Dr. B. Espinet. This is a hospital over in Barcelona, Spain. We can see that Eddie Dubois is from the University of Groningen, that is in the Netherlands, and apparently it's one of the most traditional and prestigious universities in the Netherlands, and it has a high reputation for academic teaching and research. So researchers are utilizing optical genome mapping. They're reporting their findings all across the internet, and I just wanted to check on Google to see if I can find anything else. And I found this newly published 20th of April 2022 Nature article, and we can see here multiplex structural variant detection by whole genome mapping and nanopore sequencing. So they were using, I think, Oxford nanopore alongside Bionano Genomics optical genome mapping, and the statement here states optical mapping can economically and quickly detect structural variants across a whole genome, but it does not provide sequence level information or precisely resolve breakpoints. And perhaps it doesn't, but what was interesting was 22 hours ago when they released this, on encephalopathy, so they detailed how Bionano Genomics optical genome mapping precisely determined breakpoints on the CDKL5 gene in a girl with this disease. What's interesting to note is that they combined optical genome mapping with a next generation high throughput sequencer, and they just gave some background and said, it's really important to map out these genomic structural variations, and that the discovery of structural variations is complicated because of the variations in their sizes. And that's why the employment of a single technology or approach for structural variation investigations is unlikely. They use multiple machines, different machines. So for the smaller structural variations, they often use these short reads, high throughput sequencing technologies. They're highly accurate, but short reads are less suited for long and complex structural variation characterizations, which is where BNGO comes in for those larger variations. And they stated in the abstract here, when they're looking at raw molecule length averaging, kilobases, 300 kilobase pairs, we can see here it enables cheaper and quicker structural variation characterization, and it's widely adopted for creating maps of long-range structural variations and complex rearrangements. But it lacks sequence level resolution, and with optical mapping alone, it's not possible to accurately pinpoint structural variation breakpoints. So that being said, if you need to use, optical genome mapping alongside sequences, it could be very, very interesting when the sequencing market is currently growing. Now, the people who use Oxford Nanopore alongside Bionano Genomics, all of these researchers are from Drexel. It just says Drexel University, triple Drexel University right there. Drexel University is a private research university with its main campus in Philadelphia. Drexel University is a world-class comprehensive R1 research institution and a global leader. Apparently R1 means doctoral universities, very high research activity. And talking about this sequencing market, we can see here that Allied Market Research currently states that the global next generation sequencing market accounted for about 6.5 billion in 2020, but they believe that the market size is going to increase by 2030 to $35.5 billion. They state that next generation sequencing is steadily being integrated into clinical laboratory analysis, testing, disease diagnostics in healthcare sectors globally. But next generation is also being widely used in pharmacogenomics, and they're trying to accelerate the drug discovery process. One thing I also wanted to show was this white paper stating that the Sapphire system, the new system, uses a high throughput optical mapping technology to detect structural variants missed by next generation sequencing systems. The speed and flexibility prove that the Sapphire is the answer for both human and translational research projects. So this is another university, the University of North Carolina School of Medicine, and it's just interesting to see more and more research coming out. For example, optical genome mapping as a diagnostic tool in pediatric acute myeloid leukemia. So they're utilizing the machine, and they're exploring a lot of different rare disease and cancers at the moment. Even more interesting, I headed over to LinkedIn and I wanted to see what was being published over there, and take a look at this. So the vice president at Bionano Genomics, Alex Hastie, was sharing Ravindra Kohli. This guy is a researcher who puts out so much research at the moment using optical genome mapping, and showing how useful optical genome mapping is. Alex Hastie said that this topic has received a lot of attention. Optical genome mapping is a very robust and simple technique for QC of your cell lines because it's genome wide and very sensitive. And if you scroll down to the post, who is tagged in it, you can see that Frederick Westerlund, he is a full professor and head of division at Chalmers University of Technology. He tagged the researcher at AstraZeneca who is currently using the machine, who is Sumaya. And I think I remember they were exploring and looking at optical genome mapping in terms of trying to develop, I think it was gene therapy or certain gene therapeutics, precision medicine. It was something like that. Next, in terms of pets, in terms of the animal world, there's a company called Vidium Animal Health, and it says here that a month ago they were utilizing optical genome mapping to reveal novel genomic features of canine osteosarcoma. So what do you reckon, guys? Do you reckon there's going to be a kind of consumer market here, or a market for the vets where they're able to make money off optical genome mapping for helping diagnose certain illnesses and diseases in animals? Because right here it seems that this is a pet cancer diagnostic company. You can see they are stating that Vidium is dedicated to unraveling the genetic components of common and complex diseases including cancer. We intend to open up the use of precision medicine to combat canine cancer in new and more effective ways. So if they're potentially looking at precision medicine to combat canine cancer, and they're using optical genome mapping to explore canine osteosarcoma, which is a malignant tumor of the bone, its most common bone tumor found in dogs. They are also looking at detection and characterization of the on and off target integration of foreign DNA in the host genomes, and they're utilizing, novel optical DNA mapping technology by Bionano Genomics for therapeutic cell and gene therapy development. And this is from the senior scientists Sumaya at the Gene Editing Safety Group at AstraZeneca. Apparently, this article from biospace.com, released in March 2022, has stated that the global precision medicine market is predicted to hit around $140.69 billion by 2028. This precision medicine is a new approach to disease prevention and management that is gaining popularity. Where will Bionano Genomics fit in all of it? We can see also that Professor of Bioinformatics at Radboud University Medical Center is using optical genome mapping to identify variants in the dark parts of the genome. This one was really interesting, reported 21 hours ago by Claudia Aipucha. I had to put it into translate because I think it's in Spanish or Portuguese. She said she wanted to share a recent talk on the methodological foundation of optical genome mapping, a technique that aims to be implemented as a complement to classical cytogenetics with G banding, and it allows to determine the presence of numerical and structural variants from the 500 base pairs upwards. So she stated some advantages and some disadvantages as well, so advantages and limitations of the machine. And in the final statement, she said, although it's still an experimental non-diagnostic stage, optical genome mapping that is, given the results obtained, we estimate that it will soon be recognized as a diagnostic technique. So what exactly is CROM, the Institute de Cytogenetica Humana? When I looked them up on their website to see where she's from, I could see that it's based over in Argentina, Buenos Aires. Another interesting LinkedIn message was from Anjali Jain. So this was a video that we watched in the other episode just before. And within this video, she was explaining stuff about optical genome mapping and asking some questions. And in part 2, we can see here Adam C. Smith was talking about part 2 video. She asked the question, is optical genome mapping ready to be clinically implemented? And he said the answer is yes. There are several labs, including the UHN Cancer Cytogenetics Lab. I think this is one of the massive ones in Toronto. It's a University Health Network lab, I believe, that have run our own internal validation studies that show that optical genome mapping will replace karyotyping, FISH, and microarray as frontline testing for many hematological malignancy indications. So lots of blood cancers, they're saying it's going to replace it, they believe, and that apparently the University Health Network has run over 200 samples on OGM and compared the results to conventional approaches. He's saying that optical genome mapping provides a level of resolution and clarity that is not achievable with conventional assays. Short read sequencing has limitations with structural variation assessment, and long range sequencing is currently quite expensive, especially at 300 to 400x required for rare variant detection. 2022 will be the year of OGM. Harpreet Kaur has said at the University Health Network, thank you for sharing this wonderful news with us, Dr. Smith. We had reviewed published literature to put this webinar together a few months ago. It's great to hear that the UHN Cancer Cytogenetics Lab has validated OGM. We agree with you that OGM has great potential. I just saw this released as well. If anyone can drop the comments below, it was about May 2022 CPT editorial panel meeting, and it included something about optical genome mapping in here too. So it stated number 25 tab at the American Medical Association. This is the proposed panel agenda, and here at number 25 they said cytogenetics with optical genome mapping. They've got a number code there, an established code 8X006 to identify optical genome mapping methods of cytogenomic analysis for chromosomal abnormalities. So I believe this is kind of like a medical code. I'm not sure what it's used for. I think in America it's different to over here because everything is free here with the NHS, but I think the medical code is for them to identify ways of diagnosing or utilizing certain medical equipment, and I think it's also for health insurance reimbursement purposes, but I'm not sure. So if you know, drop me some comments down below how it works. Teach me about how it works in America. On the other side of things, we can see that the McDonald Genome Institution is actually utilizing the Sapphire system because they're offering whole optical genome mapping services with the Sapphire. Apparently as well, if you take a look at Verve Therapeutics, they're looking for kind of precision genome engineering, and apparently they're using whole genome optical mapping also. So who exactly are Verve Therapeutics? They're a biotechnology company that have a singular focus. They're created to protect the world from heart disease. So they're trying to pioneer single course genetic editing medicines to treat cardiovascular disease. If you pop this open as well, in Australia they're using Hi-C, I think it's PacB's Hi-C, and optical genome mapping alongside to look at the genetic and genomic resources for soybean breeding. I can't open this file because I won't be able to film while I open a PDF document for some reason. You can also see the Oxford Nanopore and Bionano Genomics technologies are being utilized together for plant structural variation detection, April 2022, and this includes researchers from the French National Institute for Agricultural, Food and Environment, INRAE. Over at King's College London, Dr. Indu Sri Rajan Babu is a visiting research associate, and her research is focusing on rare disease diagnosis using next generation and long read sequencing and optical genome mapping technologies. Over at Oxford Brookes University, very interesting, they're looking for a research fellow. They want to pay them around 32k to 36k pounds, grade 8 to 9 per annum, and this was placed on the 27th of April, so it's a newly put out job. And what are they looking for over here? The post will involve working with a team to run an optical gene mapping platform, Sapphire system, Bionano Genomics, as well as utilizing whole genome sequencing and whole exome sequencing data from a large cohort of patients with developmental eye anomalies to identify novel genes and pathways for these disorders, underlying these disorders. And check this out, in addition to performing innovative research, this post provides an opportunity to directly benefit the families affected by these life-changing conditions by contributing to our ongoing translational work to enhance clinical genetic testing in collaboration with our NHS partners. So lastly, while Bingo is dropping, what's the stock now? $1.50. While Bingo continues to drop, I'm going to continue to accumulate. In the long run, I believe in this company a lot. I know they've got some innovative technology, but we need to see how they play the game, how their business builds out, how they manage to market the product and continue to get those sales. Thank you very much for watching. Please remember, none of this is financial advice, it's for entertainment only. Please hit the like button, hit that subscribe button, and I'm gonna catch you in the next video. Mr. Investor, over and out, baby.